Help me welcome Matteo Pigni.
Hello everybody! I would like to start by thanking all of you for being here. I'm glad to see many faces that I actually don't know. I expected to see a lot of people from the department, but actually many of you I don't know, so thank you for being here. I call you new friends. I'm going to be talking about experimental models in immunology. Experimental models in immunology and how we can use these experimental models to understand better how infections and immunological disorder works and how we can fight these infection and immunological disorders through these models. As Dimitri said, this talk, the most part of the work that I'm going to show you comes from my PhD thesis, and here you see the front page of my thesis with all the people that were in the jury and everyone. This is me. And this is the title of my thesis: Generation and Utilization of In Vitro Models of Dendritic Cells for the Study of Dendritic Cell Biology. And I wanted to show you this because I think that in the title of my thesis there are two key elements to understand the work that we've done. The first element, as we said, is the concept of model, and the second element is this group of cells that are immune cells known as dendritic cells. And I'm going to show you what a model is, what dendritic cells are, and then we're going to go through how we generated this model of dendritic cells and what this model is good for. And I would like to start by talking about the concept of model, which is not necessarily restricted, as many could think, to hard sciences, but actually it's a concept that is intrinsic in many fields of human knowledge, because models reflect somehow the way that our brain uses to collect information and process information and give a structure to information. And we have several examples of models in everyday life also. We know, for example, architects use models. They don't have just constructions built to look at them and say, no, I don't like it, just destroy it, we'll start over. Or medical students, they use models to train, to learn how to treat patients, and I'm happy that nobody would lay a hand on me before training with a model. And we can find many other examples. We can find examples in fashion, in chemistry, in physics, in engineering. And what is important for models is that they all share some characteristics, that are the fact that they are simple enough to exclude all the elements of the problem that are not necessary to solve the problem, but at the same time they're relevant enough to be representative of the object of study, and they're able to allow us to generate new knowledge about the subject of study. And this is important because if any of these elements is missing, then we end up with a wrong model. And the wrong model mostly drives us to wrong conclusions. Now, I said that the dendritic cells are immune cells. And to understand the function of dendritic cells, we'll need to go through how the immune system works. You know that we're constantly exposed to pathogens like bacteria, viruses, fungi, parasites, and environmental agents. And these ones can damage our tissues and cause pathogenic processes. But fortunately, our body has barriers like skin, mucus, cilia, so on. And these barriers normally manage to keep these pathogens outside, and so we stay healthy. But when the pathogens manage to overcome these barriers, then we become sick. And this is why we have an immune system which has the purpose to fight the pathogens that manage to enter the body. As you see here, the immune system comprises several different types of cells. And these cells can be divided into two large groups, the innate immune cells and the adaptive immune cells. And these two groups reflect the two branches of the immune response. There is an innate response, which is rather unspecific, but it's very fast. It responds very quickly to the presence of pathogens. And on the other side, we have the adaptive immune response, which is slower, but generates responses that are extremely targeted against each single pathogen. The dendritic cells that we were talking about are, by definition, innate immune cells. But they represent, thanks to their function that we're going to talk about, a connection between the innate immunity and the adaptive immunity. We can use an analogy to understand better these concepts. We can imagine the pathogens as criminals who want to harm our body, and the immune defenses as the group of security forces that have the purpose to defend the body from the criminals. And we have the innate immune system that can be seen as a military branch, which is very active on the field, very reactive to the presence of pathogens. And we can see the adaptive immune system as the intelligence of our body, which takes longer time to respond, but develops targeted strategies. The dendritic cells that are here in the middle can be imagined as an investigative department, and they have the role to collect information in the field where you find the military branch and bring this information to the intelligence. And the role of the intelligence is that to develop strategies to contain and clear the criminals, or also to support the military branch. But the cells of the immune system are not sufficient alone to give us protection from pathogens. But actually they need a series of structures and organs that support their function. And these structures and organs constitute the lymphatic system. This lymphatic system comprises several organs that are linked to each other and to the periphery of the body through a net of lymphatic vessels. And we're going to focus on two organs of the lymphatic system, which are the lymph nodes and the spleen. And these two organs can be imagined as the immune system's operating centers, where the T cells and the B cells, so the intelligence, reside and decide all the strategies to fight the criminals, the pathogens. Now, the function of dendritic cells is to go around in the periphery of the body and check for the presence of pathogens. And as soon as they find a pathogen, they collect evidence of the presence of this pathogen, and we call this evidence antigen. And then they can enter into the lymphatic system to migrate to the closest lymph nodes. So they travel through the lymphatic system, they reach the lymph node, and here they give this evidence and information to the T cells to fight the pathogen. Now the next step is that the T cell can use this information to identify the pathogen. So they analyze the evidence, they recognize who the pathogen is, and then together also with the B cells they can develop the better strategy to fight the criminal. And this leads us to two results. The first result, as we said, is adaptive response. So direct elimination and or containment of the pathogens, and in the meantime support of the innate immunity. And on the other side we have immune memory, which means that now T and B cells know a good strategy to fight that specific pathogen, so if we encounter again this same pathogen, they will be able to react faster and more stronger. So we have seen that the role of dendritic cells is to connect the innate and the adaptive immunity, and to direct the development of an appropriate immune response. And this is important to us, this is interesting, because if we know how dendritic cells work, we can exploit their mechanisms to generate better vaccines, or to develop better strategies for cancer immunotherapy, for treatment of autoimmune diseases, and many else. But there is a problem. Dendritic cells are difficult to study. And I'm going now to illustrate to you why they are difficult to study. I think many of you are familiar with this kids game. You know how it goes. You have a very crowded place, it's very colorful, and you know that in the middle of the crowd somewhere there is this guy, Wally or Waldo, it depends on where you are in the world, it's called a different name. But you know how to find Waldo or Wally because you know he wears glasses, you know he has this striped red and white shirt and this hat. So if you look close enough, eventually you manage to find the guy. Now, if we take a look in the spleen of a mouse, we have a very similar situation. This is like looking at the beach that we were looking earlier with Wally. This is the inside of the spleen of a mouse. And here every dot represents one cell. So there is a huge crowd in here. But we know that in the middle here somewhere there are some dendritic cells. And we know that they have some characteristics like Wally did that can allow us to recognize them. So we analyze the presence on their surface of these markers, these molecules, and we can find this small population here, which are dendritic cells. And you see here, 1–2%. This is the fraction of dendritic cells that you find in the spleen of one mouse. Meaning that for one mouse, you can get a few 100,000 cells if you're lucky. And this is not barely enough to do any experiments. So one of the big problems with dendritic cells is they're scarce in vivo. The other problem is, even if you manage to isolate enough cells for your experiments, you go in vitro, you put them in culture, and after few hours they start suffering, they don't like it at all, they start dying, and they start losing their functional properties. So, this is why it would be very useful to have a model of dendritic cells, which, as we said, with a model you can remove all the elements that are not necessary for the solution of a problem. And this instability is not necessary, but actually it's a problem for us. So we decided in the lab to generate a dendritic cell line as a model to study dendritic cells. To do this, we have taken a transgene, this SB40-T, which is an oncogene. As we said earlier in the previous talk, oncogenes can do something to cells. They have an effect on cells and also on the ability of the cells to survive. So we generate this mouse model with a transgene, and if we go to look in the spleen after 4 or 8 months of age of the mouse, we see that these dendritic cells transform and become tumoral, which of course is not great for the mouse, but it's good for us because now the dendritic cells that are tumoral are immortalized. This means that we can put them in culture, and here they will be stable, and we can expand them as much as we want. So this is how we generated our dendritic cell line. We wanted, after generating the cells, to share this with the community, because in science very much is about sharing. But to do this, to have people trusting our work, we needed to demonstrate to them that these cells that we have generated are equivalent to fresh dendritic cells. So to do this, we published this paper in which we compared the two, and we demonstrated that they look the same and that they behave the same as fresh dendritic cells. So how did we do to demonstrate that they look the same, I need to do another analogy to show you this. Let's say we have a policeman and we have all these pieces of equipment. And we know that the policeman might or might not have these pieces of equipment. We have these graphs here that tell us, no, he doesn't, yes, he does. Okay? So, for example, if we take the transceiver, we know that the policeman does have a transceiver. And we can do the same game with all the others. So, he has no axe, no hose, but he has handcuffs and a gun. If we take a fireman, we know that he has a transceiver as well. He has an axe and a hose, but no handcuffs and no gun. And what is interesting from this is that if we now have this unknown guy, but we know the profile of equipment that he wears, then we can go back, compare it to other known profiles, and find out what his job is. So, if we compare this, we see that this profile is the same as the policeman, and so we can conclude this guy is a policeman. Now, with our cells, we did the same kind of reasoning. We took the cell line, we took the fresh dendritic cells, and we looked at a series of molecules that are present or not on their surface. And as you see from this figure, each time we did a comparison, we found exactly the same profile everywhere. This means that our cells are equivalent, at least in the way they look, to fresh dendritic cells. So, look the same, done. Behave the same way. How do we demonstrate this? We have said that dendritic cells behave as detectives. So, we put them in culture, we gave them evidence, antigens, we let them the time to collect this evidence, and then we added T cells, the intelligence, we let them the time to interact with the dendritic cells, and then we went to analyze the results, that are the percentage of activation of T cells as a function of antigen concentration, which can be seen as the degree of alertness of the T cells as a function of the quantity of evidence that the dendritic cells were able to show them. And as expected, the more you give them evidence, the more they're able to alert T cells of the presence of a pathogen. So also behave the same, functional, done, it works. So now we have a good model to study dendritic cells. What can we do with this model? I will give you two examples of projects that are currently running in the lab. The first one is the study of the mechanism that dendritic cells use to feel the presence of pathogens. We know that dendritic cells, fresh dendritic cells, and other immune cells have receptors that allow them to recognize the presence of pathogens. If one of these receptors become activated, it induces a signaling that leads to the generation of a kind of an alarm in the cells. Now, if another receptor is activated, also this one will induce an activation of a pathway that will lead to the alarm. But this is a different alarm. Now, if we have the two receptors activated at the same time, what happens is that they generate a cooperative response, which is much stronger than the sum of the responses generated by the single receptors. And this effect is known as synergy, and we don't know how it works. So we wanted to understand how it works. And to understand what we did, or we decided to do, I need you to imagine the cell as a very complex machine that contains a lot of small mechanisms, and each of these mechanisms is composed of gears, which in the analogy are genes. So every gear is a gene. And we know that each of these mechanisms is responsible for controlling some functions of the cells like metabolism, repair of damage, pathogen sensing, but in many cases we don't know what mechanism controls what effect, and we don't know if these mechanisms are able to interact with each other. So, what we did was to apply an approach which is very common in molecular biology that can be summarized as break it and see what happens. We decided to take out one of these gears at a time and see if we could have an effect on synergy. And to do this, we used a system that I think many of you might know. It's the CRISPR-Cas9 system, which can be imagined as a set of tools that allow us to target specifically one gear at a time, one gene at a time. So we can take, for example, cell 1, we use CRISPR, we remove this gear, and we see no effect on synergy. We repeat the process, and we wanted to do it 20,000 times. This is why having a good model, easy model to culture, was very useful in this project. So now we go to cell N, whatever N is, and we find that in this case we remove this gear and we have a loss of synergy. So the idea is we're happy, we can go on and analyze what the function of this gene, of this gear, is in the mechanism. And you might be asking, what's the point of this? Why is it interesting? This is interesting because if we know how the dendritic cells sense the pathogens and respond to pathogens, we can use this knowledge to create better vaccines, more precise vaccines, and we can use it to help the immune system to develop a good response to infectious diseases. The second project is about autoimmunity, and I would like to take as an example multiple sclerosis. Multiple sclerosis is an autoimmune disease that targets neurons, that destroys eventually neurons. And we have a model for multiple sclerosis which is called EAE, which is experimental autoimmune encephalomyelitis. And this is a model in mouse. So, you know that normally when there is no disease going on, the neurons are recognized by the immune system as part of our body. So the immune system manages to preserve the integrity of neurons. But when there is multiple sclerosis, what happens is that the immune system believes for some reason that the neuron is dangerous for the cells, for the body. And so they attack the neuron. We could imagine this as a judiciary mistake in which an innocent guy is accused of a crime that he didn't commit. If only we could have someone brave enough to find a good, honest detective and try to clear the name of the neuron. And in this case, this brave guy is us. We have access to evidence to recognize the neuron, and we have access to information. And this information is information about the innocence of the neuron, and out of the analogy, this information is a gene that teaches to the dendritic cells how to produce regulatory signals that can modulate the activity of the immune system. So the idea is to give to the dendritic cells these elements and then go back to the mouse where the immune system is attacking the neuron and just introduce the dendritic cells to hope that they manage to convince the immune system that the neuron is innocent. And so they can go back, take everything back to normal. And this was my last slide, so this is just to show you How many people are involved in a lab, in a thesis? Many people work on these kind of things. And this is just a picture of my colleagues. This is my boss, Sasha Ornea. And I would like to thank you for your attention.
Thank you very much.